you that there's more than DNA sequence and that we should also look at DNA shape, which I show here. So the DNA is actually a double helix, a three-dimensional object, and proteins recognize DNA as a three-dimensional object. And what I'm showing here is a nucleosome when DNA is wrapped around the histone core, and transcription factors would usually bind in regions that you see here. And you can see that there are nuances or variations in DNA shape. And my talk, I want to convince you in my talk that transcription factors care about these nuances in DNA shape. I want to show you here the model of the Watson-Crick double helix, and my talk will focus on the minor groove, which you see here in dark gray, and the color coding will be that dark gray is for concave surfaces, green for convex surfaces, and when the minor groove changes shape, the coloring of the gray will change. I want to show you this movie here, short movie. The red mesh that I added here to the double helix is an isopotential surface of -5 kT. And even though the chemistry of the double helix does not change, but the shape changes like a clay model, you can see that it affects the electrostatic potential in the minor groove. When the groove is narrow, this red mesh extends further outward the groove, and when the groove is wide, it recedes. And transcription factors very often use arginines, which are positively charged residues. DNA is negatively charged. So these variations are very important for achieving specificity. We have looked at all crystal structures of protein DNA complexes in the protein data bank, about 1,000 structures, and we have seen that many different families of transcription factors use this mechanism of that we call DNA shape readout. What I'm showing here, 4 different examples, and I show only the residues that contact the minor groove. And these are arginines, and you can see by the dark color, by the dark gray, that these are regions of narrow minor groove. And what these busy plots are here, these are 3 different entities. The blue plot is the minor groove width, the minimum distance across the groove when the protein is bound. The green plot, there are very few crystal structures of naked DNA or unbound DNA. So the green plot are predictions for unbound DNA binding sites predicted with a method that I have developed over the last few years, which is a Monte Carlo method for predicting three-dimensional DNA structure only using DNA sequence as an input model. And the red plot is electrostatic potential in the center of the minor groove where these arginines bind. And what we found here that first of all, DNA shape is very often already present before the protein binds. So it's an intrinsic property that is recognized by the protein. And in addition, minor groove width or DNA shape correlates highly with electrostatic potential in the minor groove. In a recent study, we looked at, with Richard Mann and Harman Bussemacher and Barry Honig's lab, we looked at how Hox proteins achieve specificity. And we used the Drosophila model. You can see here 8 Hox proteins that encode the development from head to toe or from anterior to posterior. And in Richard Mann's and Harman Bussemacher's lab, a pipeline was developed, an experimental computational pipeline that is called Select-Seq, that uses the full transcription factor complex, here the Hox protein with its cofactor EXD, and derives motifs that are selected by the different Hox proteins in complex with EXD. We found that a number of different motifs were selected, and we were able to calculate relative affinities, which we call specificity fingerprints. And you can see here, for instance, that labial, the first Hox protein, likes to bind the green motif, which is shown here. And that DFD and SCR like to bind the blue motif, and UBX, for instance, likes to bind the red motif. Our hypothesis, based on previous work, was that those factors differentiate between these different motifs, also using DNA shape readout. So what we did in my lab, Tiangying Zhu, a student of mine, developed a method for the high throughput prediction of DNA shape for whole genomes. And this is a relatively simple model, but it uses previously generated Monte Carlo predictions for thousands of DNA sequences, transcription factor binding sites, and then uses a 5-mer or pentamer model to data mine those predictions in a way that each pentamer shown here determines the 3D structure in its center. And then any length of DNA sequence or any number of DNA sequences can be predicted very rapidly by data mining based on pentamers. And this is a very fast approach. The whole yeast genome can be predicted in 20 seconds on a regular laptop. Using this, we found, based on the CELEC-seq data, by predicting about 650,000 sequences, and here are the 8 Hox proteins from anterior to posterior, and this heat map encodes minor groove width. Dark green is narrow minor groove, and white is wide minor groove. And we found that the 4 anterior Hox proteins have 2 narrow regions in the center of the core motif, whereas the posterior Hox proteins have only 1 narrow region in this region, and here a widening of the minor groove. And we showed with Pearson correlations that these different shape patterns that were selected are distinct. In a previous paper that we published in 2007, we showed that based on crystal structures, the SCR binding site, which is an anterior Hox protein, has 2 narrow regions. And these narrow regions attract arginines. And we have shown in vitro and in vivo experiments with Richard Mann's lab that these side chains are carrying in vivo specificity. So now, based on this prediction, we find the same result based on many more sequences. In contrast, a Hox consensus site that is essentially similar to the UBX site, UBX is a posterior Hox protein, showed in a crystal structure only 1 narrow region. And this is also shown here now, based on many sequences. And in this crystal structure, arginine 3 and histidine -12 did not bind the minor groove. And these are the residues that carried the specificity for SCR. Using those profiles, we calculated a dendrogram, not using sequence, just using minor groove width. And we got an ordering of the Hox proteins in 2 branches, an anterior branch and a posterior branch. And seeing this, we thought that this might indicate how Hox binding sites have differentiated. We, of course, looked at other properties, like here, the major groove, but the major groove width does not show this distinction. The reason for this, this figure here on the right, it shows the different functional groups of the bases on top in the minor groove and on the bottom in the major groove. Red and blue are hydrogen bond acceptors and hydrogen bond donors. And let's first look at the major groove. You see there's a distinct signature for each of the 4 base pairs in the major groove. And this is what we are seeing, that transcription factors usually contact the major groove and form hydrogen bonds. But this is often not enough for specificity. Contacts to the minor groove are necessary, and in the minor groove, the GC base pair cannot be distinguished from the CG base pair, and the AT is identical to the TA base pair in terms of functional groups. So here, DNA shape comes into play, and these distinctions in electrostatic potential are recognized and lead to specificity. Another example will be presented in the next talk. So what I showed here for Hox proteins, shape distinctions due to variations in the core motif are important for specificity. Raluca Gordon will talk about basic helix loop helix factor, where we showed that variations in the flanking sequences lead to this specificity between binding sites in various locations in the genome. And I've talked so far about transcription factors. Let me show one more example. The endonuclease DNase 1 has a much lower sequence specificity. Here's a crystal structure shown where the protein binds to the minor groove with a loop that intrudes the minor groove, and there are various 2 arginines that intrude the minor groove in this region where we have 2 blue base pairs. In collaboration with Harman Bussemaker's lab and John Stamm's lab, we measured a DNase cut rate for all 4,092 unique hexamers, and we found that in the blue positions, shown here in blue, DNase cut rate increases when the groove is narrower. And this can be explained with what I've shown before. The groove gets narrower, so these arginines get better attracted to the minor groove. And in these other 2 positions shown in green, the DNase cut rate increases when the groove is wider. You might wonder, delta G is proportional to a minus log of the cut rate. So we have shown that DNase 1 can be used as a tool to probe DNA shape. Sorry. And last application that I want to show, so we are now analyzing Snips, and we found with our new high throughput method that there are Snips that don't affect shape, like shown here. These 4 different colors are the 4 different base pairs in the central position where you see an N. And there are Snips that affect shape dramatically, like shown here. So we call them shape-insensitive versus shape-sensitive Snips. And we looked in collaboration with Angela De Paz and Sergei Nustin at non-coding regions, at cis-regulatory modules, and distinguished a set of SNPs in transcription factor binding sites or their direct flanks and versus SNPs that are not located in transcription factor binding sites. For the positive group, in transcription factor binding sites or the direct 5 base pair flanks, we found that in blue, the low frequency SNPs affect shape more dramatically. Whereas we don't see this distinction in a set that does not contain transcription factor binding sites. And we could not explain this signal only based on sequence. So here, shape seems to disrupt the protein binding. And what we are working on now to provide this to the community, we are predicting various features of shape. I only talked about minor and major groove, but other features. And we want to provide this in a format as a genome browser so that everybody can look at different data sets. One last thing, we're also building a database of 3D characteristics of transcription factor binding sites. One approach is that we use, instead of a 4-letter alphabet, we use an 8-letter alphabet where the lowercase letters characterize regions where adenine, cytosine, guanine, or thymine are located in regions of narrow minor groove, and the capital letters in regions of wide minor groove. And using this, we are building a very extensive transcription factor binding site database, and we're also using this in collaboration with Bill Noble and Yael Mandel-Gutfreund for implementing new motif search algorithms. And I want to summarize. So this figure here shows again the first story that I showed you, that a Hox protein in cyan needs a cofactor shown in yellow to reach its full binding specificity, which includes a readout of DNA shape or minor groove width. And I hope I convinced you that DNA shape matters. DNA shape information is now available at the blink of an eye on a genome-wide basis. And if you have data sets that you would like us to look at, you're welcome to talk to us. And DNA structure and sequence analysis can now be combined. And since this work requires teamwork, so you have here again the Hox protein and its cofactor reading the DNA. This work is a collaboration with various labs that are listed here. And most of all, I want to thank my own students, and some are here in the audience. And Tian Ying is here. He has developed a high-throughput DNA shape prediction server, and Iris worked on the Hox analysis. Thank you very much.
I have a quick question. Please use the microphone if you got it. So I'm wondering, is it possible that some factors are in fact functional with two different shapes that actually confer different functions? So
could be, yeah, could be.
Have you found any examples?
No, we haven't found any of them. What we are just starting. So what we are first looking at is if there is the same shape realized by different sequences. And if one is functional or the other is functional, or if both are functional. So we first look at similarities, and the next step will be looking at differences.
So I mean, it is possible that we would even discover the shape component if it is in fact mixed between different ones, right?
Right. Yeah. Right. So we will be looking at it, and we're also developing multivariate models where we clearly identify which has a larger component, sequence or shape, and if it is shape, then which component and shape. So is it minor group, major group, or any other parameter? Thank you very much. Yeah.
Thank you. Okay, next up is Antonio Gomez. So again, I have to